everybody. I'm here today to tell you a little bit about membranes and why we're interested in them. So this is an example of a membrane. It keeps what's inside the cell inside and what's outside the cell outside, and we're interested in the purple blobs that lie in it. These are membrane proteins. And there's a number of reasons why we're interested in them. We want to know what they look like, their structure, we want to know their function, what they do, we want to know what other proteins they interact with and what effects that these might have. And we care about all of this because we can use this information to try and come up with new drugs for various diseases. And some of the diseases that we're looking at include Parkinson's disease, they include rheumatoid arthritis, they include a genetic form of obesity, and they also include a diabetes protein target. So even though the protein target is only the first step in the long pipeline, finding a new drug can take anything between about 20 years and about $1 billion. So it's a difficult challenge, and we try to come up with new techniques to help us along the pathway. So chemical space is very vast. There's about 10 to 60 possible compounds out there. Maybe about 10 to 40 of them are actually what we call drug-like, and much smaller number are actually what we call synthetically feasible. But there's plenty of options out there. So how do we go from all of this chemical space to the one-hit drug? And even if we know what the protein target is, which we do in our case, the membrane protein, how do we find that elusive needle in the haystack? Okay, there's still plenty of compounds that might bind to our protein, but they mightn't have the effect that we want. So we use a number of techniques to try and reduce the cost and the time in this process. And it's all based around a simple concept of a lock and key. Okay, so a protein has a region, which is the lock or a binding site, and the drug or the small molecule that will fit best into that binding site is the key. Okay? So if you take the protein structure, there's a little groove there, it's the binding site in the blue blob, and then the ligand, which is a small molecule, and you can get software which will put the two together and orientate them together and optimize the interactions between the two. And we can use this software to try and come up with new drugs that might interact better with the protein and have the desired effect for the drug. Now you might recognize what that is, but I'm sure most of you have probably had some of it today already. It's caffeine, okay? So I'm going to give you an example to illustrate the process. Caffeine binds to a membrane protein known as the adenosine receptor, and it's a member of a family of G protein coupled receptors, or GPCRs. Hugely important family because about 50% of marketed drugs target one of these. And we're lucky that we know a little bit about the structure, what the protein looks like, and where the small molecule will interact with the protein will bind. And this is just zoomed into the interaction site. And this is what the small molecule will look like in its protein. So we know what kind of shape it has to be, what kind of interactions it has to have, and we can use this information to try and come up with new compounds that will work better, new drugs. Okay? And we're interested in caffeine and the adenosine receptor because it has been linked to Parkinson's disease. Men who drink coffee are about 5 times less likely to develop Parkinson's disease, and it's just linked to increased levels of dopamine, which help prevent Parkinson's disease. Yeah. So, in the lab, we're a biology lab, so we make this protein in our cells, and then we come up with our compounds that we've designed, and we test them to see if they bind, and see if they interact, and see if they have the right effect. And then we talk to our lovely chemistry friends, and they make lots of variations of the small molecule that we're interested in to just try and tweak it and optimize its interactions and come up with a better drug for us. And then we go through lots and lots and lots of steps, and hopefully we get to a stage where we optimize and tweak the situation. We take our drug, we refine it, we test it again, we talk to our chemistry friends, we loop and loop and loop around, and we try and improve things like its safety and its toxicology and all those kind of things. And then hopefully, eventually, it would be great if we got into a stage where we can test these compounds on humans in clinical trials and see what they actually really do in real life. We're not there yet, but it's, you might as well aim high. Obviously, we're in academic labs, so we can be a little bit resource challenged sometimes. So we can talk to pharmacy and, pharmaceuticals, and try and get their help on some of these steps on the pipeline. Clinical trials are obviously very expensive, $500 million. But there's also a very, very high attrition rate as we go through this pipeline, and very few drugs actually ever make it to the market. But what we would hope at some stage is that we would, one, have a situation like this, where we have a drug that might benefit humans for various diseases, and maybe if it's a blockbuster, we might actually make a little bit of money on the side as well, which would always be nice. I put this slide up because obviously this kind of work is very multidisciplinary, and I want to thank a few of the people that are involved. There's some of the current members of the membrane protein lab, and some of our chemistry people, and you have to thank all of the sponsors who helped give us some money. So that was kind of a whistle-stop tour of some of the stuff that we do, but just want to say thanks for listening. I hope you know a little bit more about what we do, and happy Christmas to everybody.